Welcome to the molecular story of acetylcholinerase, the enzyme responsible for the termination of signaling between our nerve cells and motor neurons through the hydrolysis of our neurotransmitter acetylcholine. We see the reaction of how this happens. Acetylcholinerase is a serine hydrolase that catalyzes acetylcholine into acetylcholine with the help of water. It does this with a mechanism that utilizes a catalytic triad similar to that of trypsin and chymotrypsin with an acidic residue, a histidine, and the polar residue serine. However, acetylcholinerase has different binding residues that target acetylcholine specifically. To see more about acetylcholinerase, the structure will now be examined. Looking at the primary structure, acetylcholinerase has 586 residues, with 62 of them being negatively charged and 50 of them being positively charged. We also have 66 aromatic residues from its composition, as highlighted in purple. In addition, it has several cysteine residues, which indicates the protein's ability for disulfide bonds. It has several hydrophobic residues underlined here, indicating the hydrophobic effect as a driver for the tertiary quaternary structures of the acetylcholinerase. Secondary structures has 12 alpha helices labeled in purple that surrounds 112 beta-stranded mixed beta-pleated sheet labeled in blue. We can see furthermore that these secondary structures are made up of hydrogen bonds related to the backbone of the protein between the carbonyl group of one amino acid and an amino group of another. As for the tertiary structure, we first start off with van der Waals interactions in the form of pi stacking between the aromatic amino acids of our structures, as you can see here. Next, we can also see various hydrogen bonds between the side chains of the residues themselves, with these examples in the blue dashed lines. Notice the residues involved being polar and sometimes charged like aspartate 397. Finally, another contributor to the folding of the tertiary structure are these three disulfide bonds identified in red within the monomer. Here we can see a specific example of such disulfide bond between cysteine 265 and cysteine 254. However, the most important characteristic of acetylcholinerase is this gorge, and in this gorge is a catalytic site of where acetylcholine will get hydrolyzed, highlighted in red. It is a steep gorge which is around 20 angstroms deep and is quite narrow around its entrance, around 5 angstroms wide. Going back to that number of aromatic and hydrophobic residues from the primary structure, the gorge is surrounded by such around its periphery, as shown by the green and pink colored residues. Noting also that there are mostly on these loops right here, which are between the main secondary structures of acetylcholinerase. There are a few other types of residues, such as the acidic amino acids of glutamate and aspartate in purple. In consideration of the amino acids that make up the gorge, the first aromatic rings would guide the hydrophilic acetylcholine into the active gorge through weak van der Waals interactions and would be trapped inside the gorge from the hydrophobic residue, such as the leucine and the glycine, since the acetylcholine is hydrophilic itself. As for the acidic residues, it can provide some guidance and conformation correction to acetylcholine through hydrogen bonding of the positive ammonium group of the acetylcholine to the negative charge R groups of the side chains before entering inside the gorge. However, these have even more concrete applications when it comes to the inhibition of acetylcholine, which we will touch upon later. When it comes to the active site of acetylcholinerase, it is important to talk about how fast the reaction is. In fact, acetylcholinerase is able to hydrolyze acetylcholine to its acetate and choline parts in 80 microseconds. With such speed, examples of acetylcholine binding to the active site and binding sites of acetylcholinerase would be very difficult to capture. Instead, what we have here is a competitive inhibitor, edrophonium, which attacks the same active site of acetylcholine. Looking at the residues involved, serine 200, right here, would bind to the carbonyl carbon of the ester group of acetylcholine. It is also one of the main components of the catalytic triad is known as the esteric site. On the other hand, we had tryptophan amino acid that would electrostatically interact with the ammonium ion of acetylcholine from its ring, known as the anionic site of acetylcholinerase. The two sites I mentioned are the binding sites of acetylcholinerase. Another site to mention is this peripheral site that has a positively charged side chain of glutamate around the entrance of the gorge and tryptophan for electrostatic pi interactions. For acetylcholine, this entrance is a transitional binding site which should guide acetylcholine deeper into the gorge, as mentioned before. Looking at this mechanism, it is very similar to that of the catalytic triad of trypsin and chymotrypsin, especially considering the residues serine, histidine, and glutamate involved. Overall, we'll see both serine as the residue for covalent catalysis, and histidine contributing to the acid-base catalysis. With glutamine in presence, it lowers the pK of histidine to act as a general acid and base. In step 1, histidine performs a general base to deprotonate serine, to proceed serine to nucleophilically attack the alkyl carbon of acetylcholine. Once the tetrahedral intermediate is made, the glycines here and alanines which stabilize the intermediate. Step 2, histidine then acts as a general acid which donates a proton to collapse the intermediate, breaking off the choline molecule. Histidine then accepts a hydrogen from water to make a hydroxy group, which binds the remaining alkyl group connected to serine. Histidine as a general acid donates protons to the serine, resulting in the collapse of the intermediate and the final release of acetate. Finally, we will discuss and recall back to our discussion on inhibition itself. The reason for such is the medical application of inhibition of acetylcholine related to any neuromuscular disorder such as myasthenia gravis, a lifelong condition resulting in a muscle weakness and fatigue. For now, we'll see the structure and function of edrophonium as a competitive inhibitor, especially looking at the substrate binding here. There is a difference of bindings to that of acetylcholine, as there are hydrogen bonds of the hydroxyl of edrophonium to histidine and serine. Here we have a reason why edrophonium is used to treat acetylcholine-related disorders. First, the structure inhibitor and acetylcholine are very similar, with positive ammonium group on one side, with a slight difference of an aqua group on the edrophonium in comparison to the ester group of acetylcholine. This is the reason for the hydrogen bond that is created on the active site of acetylcholinerase with edrophonium. To see the effect of edrophonium inhibition properties, we'll see kinetic plots for the relationship of acetylcholine and acetylcholinerase with and without the presence of edrophonium. With edrophonium, the Km increases, which indicates a lower affinity to acetylcholine as seen here. However, Vmax is also lowered, which contradicts the statement that edrophonium is truly competitive and does not fall under any of the definitions of typical classifications of inhibitors. Usually with competitive inhibitors, high substrate concentration of acetylcholine would eventually overcome the increased Km. With the lower Vmax, that solution is not possible, making the inhibitor even more effective against acetylcholine catalysis. The key takeaway of acetylcholinerase here is the properties of the active gorge. Especially with the residues in both the catalytic site and the peripheral site, they provide opportunities of inhibition. The peripheral site showing effective non-competitive allosteric inhibition in its place. As we have seen, to treat neuromuscular deficiencies, the structure of both the substrate and enzyme are key into making effective inhibitors.